Hello everyone. Welcome to the new session of Respiration in Plants. Okay, so today we are going to solve the NCERT exercise from the chapter Respiration in Plants. Okay, so let us start the session. And here comes the first exercise question. So, what is the first question? Differentiate between the A, respiration and combustion. We have to differentiate between the respiration and combustion. B, glycolysis and Krebs cycle. It means that what is the difference between the glycolysis and Krebs cycle process. Then the third will be aerobic respiration and fermentations. Okay, so let us see the first difference, that is between the respiration and combustion. So column wise difference is given over here. So the respiration and combustion difference. Okay, so it is a biochemical process. So it is a biochemical process because in the presence of enzyme, many biochemical reactions takes place. So it is a biochemical process. Where is a combustion? It is a physiochemical process. Okay, so no, like enzyme is responsible here for the going on to this physiochemical process. Okay, so it's something, it is burned and it is a combustion, so it is a physiochemical process. It is not any biochemical process. It is not like involved any type of enzymes to overgo this reaction. Whereas in the case of respiration, respiration is a fully a biochemical process. Okay. Okay, next is the it occur in the living cells. So where it is occurring, this all biochemical reactions taking place in the inside the cell only. Okay, inside the cell, inside the cytoplasm, half of the part of the cellular respiration takes place in the cytoplasm itself, that is the glycolysis process, and later the other half of the reactions that take place inside the mitochondria, right? Mitochondria. So whereas this whole process is occurring inside the living cells only, and in the combustion it does not occur in the living cells. So living cell is not actually responsible for this combustion process, right? Okay, the third difference is the ATP is generated. Now in this reaction, the biochemical we can say that biochemical energy currency is synthesized in this process, cellular respiration, and it is generated in the form of ATP. Energy is generated in the form of ATP, whereas ATP is not generated in the combustion process. There is no ATP generation in the combustion process. Enzymes are required in the respiration process. There are so many enzymes. Okay, so many enzymes that are responsible for the cellular respiration. So enzymes are required in this process, whereas in the combustion no enzymes are required. Okay, the last difference, it is a biologically controlled process. Okay, so this process is a biologically controlled, whereas it is an uncontrolled process. Combustion is not going to be a controlled process. Okay, so this is the difference between the respiration and combustion. Now moving to the next difference, that is the glycolysis and the Krebs cycle. What is the difference between the glycolysis and the Krebs cycles? So it is a linear process. Glycolysis is a linear process. It starts from the glucose and ends to the that is a 6 carbon compound, right? 6 carbon compound, and in the process of glycolysis, the end product will be the pyruvic acid, which is a 2 carbon compound. So 2 carbon dioxide is lost in this process, and it is a linear process. Glycolysis is a linear process, whereas a Krebs cycle that is occurring inside the mitochondria, it is a cyclic process. It is a cyclic process, right? Okay. Next difference, it occur in the cell cytoplasm where it is occurring. The glycolysis process takes place in the cytoplasm of the cell. Okay, it is occurring in the cytoplasm of the cell. Whereas the Krebs cycle, it occur in the mitochondrial matrix. So mitochondria is having its own matrix, that is the liquid substance. Okay, so this mitochondrial matrix is there, and the Krebs cycle is taking place in this mitochondrial matrix only. Okay. So this is the major difference between this glycolysis and the Krebs cycle. Another, it occurs in both aerobic and anaerobic respiration. So this is one of the common reactions that is occurring inside the cytoplasm. That is whether it is an aerobic reaction that occur in the presence of oxygen. Okay, oxygen is there. Whereas in anaerobic respiration, where there is no oxygen is there. So both the type of this respiration, glycolysis is common. The end product, that is the pyruvic acid, that is going to be a common, whether it is a aerobic or anaerobic reactions. Now, it occur in aerobic respiration. The Krebs cycle is occurring only in the aerobic respiration. It is not occurring in the anaerobic respiration. Okay. Next, it generates two NADH2, two ATP molecules on the breakdown of one glucose molecule. So this is the net gain. Net gain that is showing. Otherwise, it is 4 ATP, but 2 ATP is used in the glycolysis process. The overall gain is the 2 ATP, right? But 2 NADH2 is also generated in the glycolysis process. So here, what it is generated, okay, glycolysis process generates 2 NADH2 and 2 ATP molecules on the breakdown of one glucose molecule. Whereas in the Krebs cycle, what it is giving 6 NADH2. Now here you can see the difference. 6 NADH2, 2 FADH2 and 2 ATP molecules on the breakdown of 2 acetyl coenzyme A molecules. So the pyruvic acid is converted to the 2 acetyl coenzyme A, right? And it produces overall the 6 NADH2, 2 FADH2 and 2 ATP molecules. So this is the difference between the glycolysis and the Krebs cycle. Now, the next difference is about the aerobic respiration and the fermentation process. So first is the oxygen is used for deriving energy. Oxygen is used for deriving energy, whereas in the fermentation occurs in the absence of oxygen. No oxygen is required in the process of fermentation. Okay, fermentation process do not require any type of oxygen. No oxygen is required. Whereas aerobic respiration, oxygen is used by deriving the energy. Next difference occur in the cytoplasm and the mitochondria where this aerobic respiration is taking place. So for aerobic respiration to take place, there must be the glycolysis process, there must be the Krebs cycle. And the glycolysis process is occurring in the cytoplasm, whereas the this Krebs cycle is occurring inside the matrix of the mitochondria. So both the organelles are very, very important. Both the compartment, that is the cytoplasm and the mitochondria. Next is the occur in the cytoplasm. So fermentation is taking place in the cytoplasm only. No mitochondria is required, or mitochondria is involved in the process of fermentation. End product are the carbon dioxide and water. So what will be the end product in the case of aerobic respiration? The end product will be the carbon dioxide and water. Whereas the end product are the ethyl alcohol and carbon dioxide. Now, in the case of fermentation process, the end products are ethyl alcohol. Ethyl alcohol. Instead of carbon dioxide, it is giving the ethyl alcohol, and the carbon dioxide is also produced. Complete oxidation of the respiratory substrates takes place. Now, in the case of aerobic respiration, it is showing the complete oxidation of the respiratory substrate that is taking place in the aerobic respiration. Whereas in the fermentation, incomplete oxidation of the respiratory substrate takes place. So here in the case of fermentation process, incomplete oxidation of the respiratory substrate takes place. So this is one of the major difference between these two. Okay. Next, about 36 ATP molecules are produced. So this is so large number, right? 36 ATP molecules are produced in anaerobic respiration, whereas in the fermentation, only 2 ATP molecules are prepared. In the fermentation process, 2 molecules are prepared. So this is the difference. This is the difference between the aerobic respiration and the fermentation. Moving to the next question, what are respiratory substrate? Name the most common respiratory substrate. So the answer is the compounds oxidized during the process of respirations are called as the respiratory substrates. So whatever the compound that is used for the process of respiration, suppose that could be the glucose, that could be the fructose, that could be the dihydroxyacetone phosphate. So whatever the substrate is there that is used for the process of respiration is called as the respiratory substrates. Okay, so carbohydrate, okay, generally the carbohydrates are there for the first preference to be the respiratory substrate. So that could be the carbohydrate, especially the glucose that is acting as a respiratory substrate. But sometimes the other food materials like of fats, proteins, and organic acids are also act as a respiratory substrate, but that is also converted to the carbohydrate. So once the fats are used, once the proteins are used, okay, it is get converted, and basically the first preference will be the carbohydrates, and then among that if it will be the glucose molecule. And if it is another type of carbohydrate, then it is first changes to the simpler form, that is the glucose. Suppose glycogen is used, so glycogen is again that could get converted to the glucose, and then it will get entered inside the glycolysis process, right? So the carbohydrate is the first preference, but yes, there are so many other nutrients that also get as a respiratory substrate. That could be the fat, proteins, and the other organic acids. Okay, next question. Give the schematic representation of glycolysis. So, glycolysis representation. So, here that is the glucose molecule. Okay, that is a 6 carbon and glucose 6 phosphate. Now, here you can see one phosphate group is get attached. So, the first reaction in this is the ATP conversion to the uh, adenosine ADP, and glucose 6 phosphate is there. So the first compound is the glucose 6 phosphate. Okay, now this glucose 6 phosphate is then converted to the fructose 6 phosphate. Okay, fructose 6 phosphate. Now this fructose 6 phosphate, okay, it is a 6 carbon compound. Earlier the this was the glucose phosphate, but here it is get converted to the another form of it, that is the isomerase enzyme is required here, the fructose 6 phosphate. Now the fructose 6,1,6 bis phosphate is changed to triose phosphate, that is the glyceraldehyde 3 phosphate. That is the two molecules. Okay, two molecules of glyceraldehyde 3 phosphate is synthesized. Okay, now this glyceraldehyde 3 phosphate triose biophosphate 1,3 bis phosphoglyceric acid is formed. Okay, the 1,3 bis phosphoglyceric acid. So this is again this triose phosphate, that is the 3 phosphoglyceric acid that is converted to the 2 phosphoglycerate. Okay, now it get two molecules of 2 phosphoglycerate by the removal of water molecule. It is converted to the 2 phosphoenol pyruvate. 2-phosphoenol pyruvate, that is the glucose is converted to the glucose 6 phosphate and ATP to ADP, right? And at the last 2 pyruvic acid is there, which is a 3 carbon compound. What are the main steps in the aerobic respiration? So the major step in the aerobic respiration. Okay, very, very important. The major steps in the aerobic respiration and the site where they occur are listed in the given below. So the glycolysis, the first, that is the cellular respiration process, is the glycolysis. Where it is occurring? The site of occurrence of the glycolysis is the cytoplasm. So in the cytoplasm, this glycolysis process is taking place. Next important step is the Krebs cycle. So that is occurring inside the matrix of the mitochondria. So all the steps of the Krebs cycle, okay, that it gets completed inside the mitochondria, and it is taking place in the matrix of the mitochondria. Okay. Another step, that is the electron transport system. So electron transport system is occurring the inner mitochondrial membrane. So at the actual situation of the electron transport system, that is occurring in the inner mitochondrial membrane. Right, mitochondrial membrane, it means that it is a double membraned organelle, right? Two membranes are there. So the inner membrane that is having a lot of transport protein that is occurring as the subunits of the electron transport system, and that is why it is a part of the inner mitochondrial membrane. Oxidative phosphorylation, very, very important. F0 and F1 particles in the inner mitochondrial membranes are there, that is used for the synthesis of the ATP molecule, and that is why it is called as oxidative phosphorylation. So again, that is the glycolysis that is occurring in the cytoplasm, Krebs cycle in the matrix of mitochondria, electron transport system occurring in inner the mitochondrial membrane, and oxidative phosphorylation that is occurring in the that is occurring at the site of this F1 particle. F1 particle that is composed of F0 and F1 particle that is embedded in the inner mitochondrial membrane. Give the schematic representation of an overall view of the Krebs cycle. So this is the Krebs cycle. So first of all, you can see this pyruvate. Okay, pyruvate, which is a 3-carbon compound. Okay, and it is converted to the acetyl coenzyme. So for this, we required a complex enzyme over here. So this NAD is converted to NADH2. Okay, carbon dioxide is released out here, and the coenzyme A is get entered in here to form the acetyl coenzyme A. So pyruvate or the pyruvic acid is converted to the acetyl coenzyme A. That is a 2-carbon compound. Acetyl coenzyme A is a 2-carbon compound. Now this acetyl coenzyme get enter inside the Krebs cycle. So from here the acetyl coenzyme, this Krebs cycle started. So the first compound, that is the oxaloacetic acid, accept the acetyl coenzyme A, okay, and it forms the citric acid. That is the 6-carbon compound. Citric acid is a 6-carbon compound. Now it eliminate out one of the carbon dioxide, and NAD is converted to NADH2, and here you can see the alpha glutaric acid. Alpha glutaric acid 5-carbon compound is formed. Alpha ketoglutaric acid 5-carbon compound is formed. That is the alpha ketoglutaric acid. Now, this alpha ketoglutaric acid in the presence of enzyme get converted to the succinate. Now, here what you see, it is the carbon dioxide that is eliminated out. NADH2 is synthesized. GDP is converted to the GTP. And succinic acid is formed. So this is a very, very important reactions that is occurring in the alpha ketoglutaric acid. The next one is the conversion of the succinic acid to the malic acid. Succinic acid to the malic acid. Now the succinic acid is a 4-carbon compound that get converted to the FAD, that get converted to the FADH2. Okay, so this is the enzyme, succinate dehydrogenase enzyme, that is attached to the inner mitochondrial membrane. Otherwise, other enzymes are present in the mitochondrial matrix, but this succinate dehydrogenase, it is embedded inside the mitochondrial inner mitochondrial membrane. Now, the malic acid that get at last converted to the oxaloacetate here, that is the 4 carbon compound, so it is also forming the 4 carbon compound. So NADH2 and NADH, that is released out here. NADH2 is formed from the NAD plus ion, so malic acid is converted to the oxaloacetic acid. So here you can see whole reaction process, that is a Krebs cycle that is occurring inside the mitochondrial matrix. Explain ETS. ETS means electron transport system. So ETS, or the electron transport system, is located in the inner mitochondrial membrane. So where is the location of the electron transport system? It is located in the inner mitochondrial membrane. Okay, all the components of the electron transport system are present on the inner mitochondrial membrane itself, and it helps in releasing and utilizing the energy stored in NADH2 and FADH2. Okay, NADH and H, which is formed during the glycolysis and the citric acid cycle. So whatever the releasing and utilizing energy is stored in the form of NADH2, FADH2, it is all the energy that is in the form of NADH2 and FADH2, it is get converted into the ATP, right, which is formed during the glycolysis and citric acid, get oxidized by NADH dehydrogenase, that is the complex 1. The electrons so generated get transferred to the ubiquinone through FMN. So here you can see the structure, okay, the inner mitochondrial membrane, inner membrane space, and this is the matrix. So here you can see it takes from the matrix NADH and it is transferred to here. Then electron is transferred to FES, then two electrons. Okay, it accepts the 2 hydrogen from the uh, matrix region, and it is get converted to the 2 hydrogen end. Then it is processed to the cytochrome B, FES, 2 electron, OH, and then to the cytochrome C and A3. So this complex 1, the complex 1 is NADH dehydrogenase. The electrons so generated get transferred to the ubiquinone through FMN. So whatever the electron is generated here, it's transferred to the ubiquinone through FMN. Okay, flavin mononucleotides. And in a similar manner, FADH2, okay, it is accepted by the complex 2 generated during the citric acid cycle. I've already told you that the citric acid cycle is only having one enzyme, that is the uh, that is the succinate dehydrogenase, right? That is the complex 2, and it is a part of the citric acid cycle. It has transferred its electrons to the ubiquinone, and the electrons from the ubiquinone are received by the cytochrome BC1 complex 3 and further get transferred to the cytochrome C. So cytochrome C act as a mobile carrier between the complex 3 and complex 4. Complex 3, and that is the cytochrome C oxidase complex, and containing the cytochrome A and A3 along with the copper centers, that is the complex 4. So here you can see the complex 4 ended. Okay. Now, during the transfer of electron from each complex, the process is accompanied by the production of ATP from ADP and inorganic phosphate by the action of ATP synthesis. So, ATP generation is very crucial step of this reaction. Okay. The amount of ATP produced depends on the molecule. So, whatever the molecules are there, it depends upon that which has been oxidized. So 2 ATP molecules are produced by the oxidation of 1 molecules of NADH. Okay, and 1 molecules of FADH2 on oxidation gives 3 ATP molecules. And if it is a 1 FADH2 molecule, then its oxidation gives 3 ATP molecule, 1.5 each. Okay, moving to our next question. Distinguish between the aerobic respiration and anaerobic respiration, glycolysis and fermentation, and the glycolysis and citric acid cycle. So aerobic respiration and anaerobic respiration, it uses oxygen for its driving energy. It uses absence of oxygen. Okay, difference between the aerobic respiration and the anaerobic respiration, it uses oxygen for deriving energy. Okay, oxygen is required for driving the energy, and here it occurs in the absence of oxygen. It means that no oxygen is required here. It occurs in the cytoplasm and mitochondria. So where the next aerobic respiration is taking place, it occurs in the cytoplasm and mitochondria. So glycolysis is taking place in the cytoplasm region, and the mitochondria is taking place inside this Krebs cycle is taking place inside the mitochondrial region. Okay. Next, anaerobic respiration. It occurs in the cytoplasm. It occurs in the cytoplasm. So no mitochondria is involved in the process of anaerobic respiration. Next, third difference is the end product of aerobic respirations are carbon dioxide and water. What will be the end product? So the end product is are the aerobic respiration are the carbon dioxide and water. What we get at the end along with the energy, so carbon dioxide and water we get at the end of the aerobic respiration. The end products of the fermentation are ethyl alcohol and carbon dioxide. Okay, so the end product of fermentation, in the process of fermentation, okay, the complete oxidation of the substrate is not taking place here. So it is converted to the ethyl alcohol and the carbon dioxide is released out. So that is the end product of the fermentation. Next, the complete oxidation of respiratory substrate takes place. Okay, so complete oxidation. Okay, so complete oxidation of respiratory, it means that no substrate is left over here. Okay, the complete oxidation of the substrate takes place, and the end product will be the carbon dioxide and water along with the energy. And here, what you see in the case of anaerobic respiration, incomplete oxidation of respiratory substrates takes place. Okay, incomplete oxidation of respiratory substrate takes place. And what you see here, that the amount of energy that is formed is quite less, and the ethyl alcohol, right? So this is not, we get the ethyl alcohol as a last end product, but that can also get oxidized later on. But here in the process of anaerobic respiration, the end product is the ethyl alcohol, and that is due to the incomplete oxidation. Now here you can see the difference between these two. 36 to 38 ATP molecules are produced in the process of aerobic respiration, whereas in the case of anaerobic respiration, only 2 molecules of ATP are produced. Only 2 molecules of ATP are produced. Next difference here, what we see here, that is the glycolysis and fermentation. So glycolysis occurring during the aerobic and anaerobic respiration. Okay, anaerobic and aerobic respiration. That is the glycolysis process. It means that whether it is a aerobic reaction or the anaerobic reaction, in the presence of oxygen or in the absence of oxygen, this glycolysis process is a very, very common process. But what happened in the case of fermentation? Fermentation is a type of anaerobic respiration. Fermentation is a type of anaerobic respiration. Right? So, and second difference you see here, the pyruvic acid is produced as its end product. Pyruvic acid is produced as its end product. Right? And second, you see the fermentation, ethanol or the lactic acid is produced as its end product. Here, the ethanol or the lactic acid, this is going to be the end product in the process of the fermentation. Okay. Next difference, what we see here is the glycolysis and the citric acid cycle. Glycolysis. So glycolysis, it is a linear pathway. You can see the linear pathway. One reaction, one enzyme is used. Second reaction, second enzyme is used. Third reaction, third enzyme is used, and so on. And it is a 10-step reaction, and you can see that every step is required. Every step is required one of the enzyme. So it is, you can see it is a linear pathway. It is a linear pathway. And what we see in the case of citric acid cycle, then it is a cyclic pathway. It is a cyclic pathway. You can see that it is going to be in the circular form, right? Next, it occur in the cell cytoplasm. So where it is occurring? It is occurring, the glycolysis process is occurring in the cell cytoplasm. It is occurring in the cytoplasm. And the citric acid cycle, that is the Krebs cycle, it is occurring in the mitochondrial matrix. It is occurring in the mitochondrial matrix. Okay, that is the liquid substance of the mitochondria. Okay, the inner compartment. Now we have seen when we are studying about the structural part of the mitochondria, then there are the two chambers. One chamber that is present between the inner membrane and the outer membrane of the mitochondria, that is the peri-mitochondrial chamber, and another one that is the formed between the inner mitochondrial membrane. So that is the inner large chamber that is having the mitochondrial matrix. So the Krebs cycle is taking place in the mitochondrial matrix. And now it occurs in both the aerobic and anaerobic respiration. So the glycolysis process is occurring in both the type of respiration, whether it is an aerobic process or anaerobic process. Now it occurs in aerobic respiration. Citric acid cycle, it occurs in aerobic respiration. Now, one glucose molecule break down to generate 2 NADH2 and 2 ATP molecule. Okay, so one glucose molecule, when it get broken down, then it generates 2 NADH2 molecules and 2 ATP molecule. 2 NADH2 and 2 ATP molecules. Okay, citric acid cycle, it produces 6 NADH2 and 2 FADH2 and 2 ATP molecule on breakdown of 2-acetyl coenzyme A. So here you can see the complete oxidation of this substrate is taking place, whereas in the glycolysis, okay, whether it is an aerobic and anaerobic process, only 2 NADH2 and 2 ATP molecules you are getting. So this is the complete oxidation and this is the incomplete one. So that is why we are seeing the difference. But this is the difference between the glycolysis and the citric acid cycle. Moving to our next question. What are the assumptions made during the calculation of the net gain of ATP? So, assumptions that we are made during the calculation of the net gain of ATP. So that is for theoretical calculation of ATP molecule, various assumptions are made, which are as follows. Now, first one, it assumed that the various parts of aerobic respiration, such as glycolysis, tricarboxylic acid cycle, and electron transport system occur in a sequential order pathway. Okay, so this is very, very important thing, that this is the sequential process. So first the glycolysis process will take place, that is occurring in the cytoplasm region. Then it get entered, the end product of the glycolysis, that is the pyruvic acid. Okay, it is entered in the first it is changed to acetyl coenzyme, then it get entered inside the TCA cycle, Krebs cycle, and then it forms the ETS, electron transport system, that occur in the sequential order, okay, in this pathway. B, NADH produced during the process of glycolysis enter into the mitochondria to undergo oxidative phosphorylation. So NADH produced during the process of glycolysis. Okay, so there are the two reactions in which this NADH2 is produced by the process of glycolysis, and it enters to mitochondria to undergo oxidative phosphorylation. Third thing, glucose molecule is assumed to be the only substrate while it is assumed that no other molecule enters the pathway at the immediate stages. So here we have already discussed about it that whatever the substrate, first of all, it is changed to the carbohydrate, and the simpler one, that is the glucose, will be the starting point of the glycolysis. So from the glucose molecule, it is assumed that this is the only substrate while assumed, and no other molecule is enters the pathway. So our calculation is based on that. For last thing, the intermediates produced during the respirations are not utilized in any other processes. Okay, so we assume that this is the uh, intermediate. Okay, there are so many intermediate that are produced during the process of respiration that is not going to be utilized in any other processes. Okay, so by this way we are going to calculate whatever the calculation we have to do in the process of energy produced during the process of respiration. Okay, now discuss the respiratory pathway is an amphibolic pathway. Very, very important question. So the respiration is generally assumed to be the catabolic process because during respiration, various substrates are broken down for deriving energy. So the respiration is generally assumed to be the catabolic process. In catabolic process, what happened? The substrate is broken down to the simpler form. Okay, and during the respiration, various substrates are broken down by deriving energy. So there are so many substrates. There are protein, there are fats. Okay, so it is broken down to derive the energy. The carbohydrates are broken down to glucose before entering the respiratory pathway. Okay, carbohydrates here, it is broken down to the glucose before entering the respiratory pathway. And fats get converted into fatty acid and glycerol, whereas the fatty acid get converted into the acetyl coenzyme before entering the respiration. So here you can see the fats are also getting entered inside the respiratory pathway. So first, the fats get converted into the fatty acid and glycerol. Okay, in the presence of lipase enzyme, the fat is broken down into the fatty acid and glycerol. Now this fatty acid get converted to the acetyl coenzyme. Okay, and you know that the acetyl coenzyme is there to get entered inside the citric acid cycle, that is the Krebs cycle. Okay, so in a similar manner, proteins are also converted into amino acids which enter the respiration after deamination. So during the synthesis of the fatty acid, acetyl coenzyme A is withdrawn from the respiratory pathway. So, okay, in the process of fatty acid synthesis, acetyl coenzyme is withdrawn from the respiratory pathway. And also in the synthesis of protein, respiratory substrates get withdrawn. So, at the time of protein synthesis, whatever the substrate, okay, this amino acid, that is also are taken away from the respiratory pathway. Thus, respiration is also involved in anabolism. Therefore, respiration can be termed as amphibolic pathway as it involves both anabolism and catabolism, right? So that is why it is better to say that a respiratory pathway is an amphibolic pathway because it is also used to synthesize some other protein and fats also, right? So sometime when the requirement is of protein and fat is more than this intermediate substances get converted into that. So that is why it is better to say the respiratory pathway is an amphibolic pathway. Okay. Okay. Here comes the next question. Define RQ. What is its value for fats? So first of all, let us understand what is the RQ, respiratory quotient. So respiratory quotient are the respiratory ratio that can be defined as the ratio of the volume of CO2 involved to the volume of O2 consumed during the respiration. Okay, it is defined as the ratio of volume of carbon dioxide, that is evolved to the volume of O2 consumed during the process of respiration. The value of respiratory quotient depend on the type of respiratory substrate. Okay, the value of respiratory quotient, okay, and it depends upon the type of respiratory substrate that is used, and its value is 1 for the carbohydrate. Okay, when you divide it by the ratio of the volume of CO2 evolved to the ratio of volume of O2 in the carbohydrate process. Okay, when you are taking the carbohydrate as a substrate, then the value is 1. However, it is always less than 1 for fats. For fats, when you divide it, you will get the value less than 1. And more oxygen for the respiration than the carbohydrate. So, this is the respiratory quotient. So here it can be illustrated through the example of tripalmitine fatty acid. So we are taking the fat, that is a tripalmitine fatty acid, which consumes 145 molecules of O2 for its oxidation. Okay, for respiration, while 102 molecule of CO2 evolved. So when you divide it by 102 by 145, the RQ value of tripalmitine is 0.7. It is 0.7. Next question. What is oxidative phosphorylation? Very, very important question. Oxidative phosphorylation is a process in which the electrons are transferred from electron donor to the oxygen. Okay, so in this process, what happened? The electrons are transferred from electron donor to the electron oxygen. Okay, whatever the electron donors are there, it is get transferred to the oxygen, which act as an electron acceptor. So here, oxygen is working as an electron acceptor. So one is reducing and another one is get, okay, oxidized. So the oxidation reduction reactions are involved in the formation of proton gradient. Okay, so in this way, whatever the proton gradient is there, there is an oxidation and reduction reaction that is taking place. The main role in oxidative phosphorylation is played by the enzyme ATP synthase, that is the complex 4. Okay, ATP synthase complex 4. Fifth, this enzyme complex consists of F0 and F1 component. ATP synthase, that is the complex 5, it is made up of the two component F0 and F1. F0 and F1. The F1 is the headpiece. F1 is the headpiece and it is a peripheral membrane protein complex and contains the site of ATP synthesis from ADP and inorganic phosphate, right? So the F1 is the headpiece and it is a peripheral membrane, the protein complex, and it contains the site of for the ATP synthesis from ADP and inorganic phosphate. Now F0 component is a part of a membrane protein complex which act as a channel for crossing of the protons from the inner mitochondrial membrane to the mitochondrial matrix, right? So when the channel is there that accepts the crossing of the proton from the inner mitochondrial membrane to the mitochondrial matrix, then for every true proton passing through the F0 and F1 complex, there is a synthesis of 1 ATP molecules. There is a synthesis of 1 ATP molecules, right? Now, what is the significance of a stepwise release of energy in respiration? So stepwise, we have to see the stepwise. Okay, so the process of aerobic respiration is divided into 4 phases: glycolysis, tricarboxylic acid cycle, electron transport system, and oxidative phosphorylation. So it is generally assumed that the process of respiration and the production of ATP in each phase take place in the stepwise manner. So this is the step what we have seen here. The first, the glycolysis occur, then the Krebs cycle or the TCA cycle will occur, and after that there is an electron transport system that occur, and at last the oxidative phosphorylation is going to take place. So it is generally assumed that the process of respiration and the production of ATP in each phase take place in a stepwise manner. So first of all, what are the steps? Glycolysis that occur in the cytoplasm. Okay, the end product will be the pyruvic acid. Now, pyruvic acid is converted to the acetyl coenzyme A, and the acetyl coenzyme A get entered inside the tricarboxylic acid cycle, that is the Krebs cycle. Now, whatever the NADH2 or FADH2 has produced in the process of glycolysis or the tricarboxylic acid cycle, that get enter inside the electron transport system. Right? And after that, oxidative phosphorylation occur. That is the synthesis of ATP. Right? So the product of one pathway forms the substrate for the other pathway, whereas the molecule produced during the respiration are involved in other biomolecular processes. Okay, so the energy that is used, that has been synthesized in the process of respiration, that is the ATP molecule, it will be used in other biochemical processes. The respiratory substrates enter and withdrawn from the pathway on the necessity. ATP get utilized wherever the required and enzymatic rates are generally controlled. Thus, stepwise release of energy makes the system more efficient in extracting and storing the energy. Okay. So with this, we have completed the NCERT exercise, the solution of NCERT exercise of this chapter. So thank you very much for attending today's session. We will meet with another session. Till then, take care and bye.